The question most people never think to ask is how. How does a repair cell in your bloodstream know that a specific tendon in your left shoulder needs attention? How does it navigate there through 300 kilometers of blood vessels? How does it know what to build when it arrives? And how does the system know when to stop?
The answer involves some of the most sophisticated molecular communication in biology. And understanding it changes how you think about everything from an ordinary cut to chronic inflammation to every emerging therapy in regenerative medicine. How your body knows exactly where to heal. Before we can understand what happens when the system activates, we need to understand the baseline state it is activating from. Most people carry a mental model of the human body as something relatively static. Organs sitting in place, cells doing their jobs quietly, systems humming along. The reality is radically different. Your body at rest is not still. It is in a state of continuous, highly regulated activity. Approximately 330 billion cells divide in your body every day. Roughly 1 million immune cells are patrolling your tissues at any given moment, not waiting for a crisis, but actively surveilling the molecular environment for anything that does not belong. That continuous surveillance is what makes the damage detection system so fast. The immune cells patrolling your tissue are not starting from zero when injury occurs. They are already present, already reading the molecular environment, already primed to respond the moment they detect a signal that does not belong. The emergency system is not switched off between emergencies. It is perpetually on standby, scanning, waiting. These tissue-resident macrophages, permanently stationed in your organs and connective tissue, rather than circulating in the bloodstream, are one of the primary early sensors of damage. They have been in the tissue for days, weeks, sometimes months. They know the local molecular environment with extraordinary precision. When something changes, when a molecule appears that should not be there, they are the first to respond.
Let's go to the moment of injury. Not as you experience it from the outside, but as your cells experience it from the inside. When tissue is damaged, several things happen simultaneously at the cellular level, and understanding each one separately is what makes the rest of the system comprehensible. First, cell membranes tear. The inside of a cell is a carefully maintained chemical environment, completely different from the outside. Proteins, DNA fragments, mitochondrial components, ATP, calcium ions, all of these exist in very different concentrations inside the cell versus outside it. The integrity of the cell membrane is what maintains that difference. When a membrane tears, from mechanical force, from heat, from chemical damage, that difference collapses. Intracellular contents flood outward into the extracellular space. These escaping molecules are biologically significant, not because of what they are, but because of where they are. ATP is the energy currency of every cell, perfectly normal inside cells, a powerful alarm signal in the extracellular space. HMGB1, high mobility group box 1, a protein involved in nuclear function, is another molecule that belongs strictly inside cells. The moment it appears outside, neighboring immune cells read it as unambiguous evidence of cellular death. Second, the extracellular matrix is disrupted. Between and around your cells exists a structural scaffolding called the extracellular matrix, ECM. It is composed of collagen, fibronectin, hyaluronin, laminin, and dozens of other structural proteins arranged in a precise architecture. When tissue is damaged, this matrix is physically torn. Enzymatic activity at the damage site cleaves matrix components into fragments. Those fragments, small pieces of collagen, hyaluronin degradation products, fibronectin fragments, carry molecular signatures that pattern recognition receptors on immune cells are specifically tuned to detect. These ECM-derived signals are particularly important because they communicate not just that a cell has been damaged, but that the structural architecture of the tissue itself has been disrupted. The scaffolding is broken. Repair is needed at the structural level, not just the cellular level. Third, oxygen drops. The blood vessels that supply oxygen to the injured tissue are often disrupted by the same force that caused the injury. Even when vessels are not directly damaged, local blood flow may be compromised by swelling and pressure. The result is localized hypoxia, low oxygen, in and around the wound zone. Cells in this hypoxic environment rapidly upregulate a transcription factor called HIF-1 alpha, hypoxia-inducible factor 1 alpha. HIF-1 alpha activates hundreds of genes involved in the damage response, including genes that produce additional VEGF, additional inflammatory cytokines, and glycolytic enzymes that allow cells to generate energy without oxygen.
All three of these categories, intracellular molecules, ECM fragments, and hypoxia signals, fall under the umbrella of damage-associated molecular patterns, DAMPs. Endogenous molecules that the immune system has evolved to read as evidence of damage, independent of any pathogen, independent of any external threat. The physics of injury generate the alarm. No external signal required. AMPs are the alarm signal, but an alarm is only useful if there are receptors sensitive enough to hear it and sophisticated enough to translate it into a coordinated response. Those receptors are called pattern recognition receptors, PRRs. They are one of the most important discoveries in modern immunology. The toll-like receptor family, TLRs, are the most extensively characterized PRRs. There are at least 10 different TLRs in humans, each tuned to recognize different molecular patterns. TLR4 recognizes lipopolysaccharide from bacterial cell walls, but also HMGB1 and certain heat shock proteins released from damaged cells. TLR9 recognizes DNA sequences characteristic of bacteria, but also unmethylated CpG sequences from mitochondrial DNA released when cells die. This dual recognition of both pathogen signals and self-damage signals is what makes the innate immune system capable of responding equally to infection and sterile injury. When a DAMP binds to a TLR, the receptor activates an intracellular signaling cascade. The most important of these is the NF-kappa B pathway, nuclear factor kappa B. NF-kappa B is a transcription factor that, when activated, moves from the cytoplasm into the nucleus and switches on genes for cytokines, chemokines, adhesion molecules, and other inflammatory mediators. The whole sequence, from DAMP binding to TLR, to NF-kappa B activation, to cytokine gene transcription, can happen in minutes. Not hours, minutes. A third signaling pathway that deserves specific mention is RAGE, the receptor for advanced glycation end products. RAGE binds to HMGB1, released from damaged cells, and amplifies the inflammatory response through a separate intracellular pathway. What makes RAGE particularly important is that it can be activated even at low DAMP concentrations. It is the sensitive early warning receptor that detects damage before the higher threshold systems are fully engaged. Together, TLRs, the NLRP3 inflammasome, and RAGE create a layered alarm system with different activation thresholds and different response profiles, ensuring that both small and large injuries are detected and responded to appropriately. Everything we have described so far, DAMP release, PRR activation, NF-kB signaling, cytokine production, takes minutes to hours to propagate through the tissue. But the body does not wait minutes before beginning to respond. Within milliseconds of cellular damage, a different signaling system is already active, one that does not require protein synthesis, one that does not require gene transcription, one that operates at the speed of ion diffusion. Calcium waves. Calcium is maintained at extremely different concentrations inside and outside cells. Outside, calcium concentrations are roughly 10,000 times higher than inside. The cell membrane is what maintains that gradient. When a membrane tears, calcium floods in rapidly, following the concentration gradient. But the initial calcium influx does more than flood the dying cell.
It triggers a cascade of calcium release from internal stores, the endoplasmic reticulum, dramatically amplifying the calcium signal. And it propagates through gap junctions, direct protein channels connecting adjacent cells, carrying the signal laterally through the tissue.
The calcium wave is not merely a passive signal. Elevated intracellular calcium activates multiple enzymes simultaneously. Phospholipases that begin modifying cell membranes, proteases that reshape the cytoskeleton, and kinases that phosphorylate dozens of target proteins in seconds. In epithelial cells at the wound edge, the cells lining surfaces like skin and gut, calcium elevation triggers immediate cytoskeletal changes that begin orienting the cells toward the wound. Before any cytokine has been produced, before any immune cell has arrived, the cells at the wound edge are already beginning to respond because the calcium wave has reached them. ERK signaling, extracellular signal-regulated kinase, is another fast-acting injury signal. ERK is a kinase that can be activated by mechanical forces as well as chemical signals, meaning that the physical stress waves propagating through tissue from an impact can directly activate ERK in cells that were not chemically exposed to damps. Injury literally travels as a mechanical wave through tissue, and ERK signaling converts that mechanical wave into a biochemical response. The body reads the physics of damage, not just the chemistry. With the alarm active and the earliest cellular responses underway, the next problem is coordination at a larger scale. The cells that need to respond to the injury, neutrophils, monocytes, stem cells, are not already at the wound site. They are circulating in the bloodstream, or stationed in distant tissue depots, or residing in bone marrow. Getting them to the exact location of the damage requires a navigation system. That navigation system is the chemokine gradient. Chemokines are a specialized subfamily of cytokines with one primary function: attracting specific cells to specific locations. There are approximately 50 different chemokines in humans. Each one binds to specific receptors on specific cell types, and each one creates a concentration gradient that cells can follow like a scent trail. The principle behind chemotaxis is straightforward, but the execution is remarkable. A cell following a chemokine gradient does not move randomly and then measure whether it got closer to the source. It continuously samples chemokine concentration across its own surface, comparing the concentration at one side of the cell to the concentration at the other side. The difference is tiny, but it is enough.
CXCL8, also known as interleukin-8, is the primary neutrophil attractant. Released by activated macrophages and endothelial cells at the wound, it creates the gradient that pulls neutrophils out of circulation and guides them to the injury site. The 2025 MedCom review confirmed that appropriate IL-8 concentrations are essential. Insufficient concentrations fail to guide regeneration, while excessive levels can paradoxically cause additional local damage. CCL2 attracts monocytes, the circulating precursors to tissue macrophages. SDF1, also known as CXCL12, attracts mesenchymal stem cells through the CXCR4 receptor, the same receptor that is the focus of significant research in stem cell mobilization for regenerative medicine. TGF-beta and PDGF gradients attract fibroblasts.
The wound site is broadcasting on 5 channels simultaneously. Each channel carries a different message to a different audience, triggering a different response. The neutrophil channel says, "Arrive immediately and clear pathogens." The stem cell channel says, "Come when the environment is ready for construction." The endothelial channel says, "Build us a blood supply." Each chemokine gradient has its own kinetics. Some gradients form within minutes. Others build over days as the healing environment matures. The sequential arrival of different cell types, neutrophils first, macrophages next, fibroblasts and stem cells later, is partly a function of which gradients form when. The injury site choreographs its own repair by controlling the timing and composition of its chemical broadcast.
The first immune cells to arrive at an injury site are neutrophils.
They typically appear within minutes to hours of damage occurring, and their behavior in that window shapes everything that follows. But getting from the bloodstream to the tissue requires navigating a barrier that normally keeps immune cells inside vessels, the vascular endothelium. This three-stage recruitment sequence, rolling, adhesion, transmigration, is not happening globally throughout the body. It is happening specifically at blood vessels in the proximity of the injury, because the endothelial cells there have been activated by the inflammatory signals from the wound. The response is geographically targeted from the beginning. Once in the tissue, neutrophils perform several functions simultaneously. They engulf pathogens and debris through phagocytosis, essentially eating cellular garbage and killing any bacteria present at the wound. They release proteases, enzymes that further break down damaged matrix components, clearing the wound bed for repair. And they deploy neutrophil extracellular traps, nets, filamentous structures of DNA and antimicrobial proteins that immobilize pathogens and amplify the local inflammatory signal. Neutrophils also release their own chemokines and signaling molecules that amplify the recruitment of additional immune cells, and critically, begin releasing the signals that will eventually call in macrophages to take over coordination of the wound response. But neutrophils are short-lived. They typically survive only a few days after activation. Their programmed death at the wound site, apoptosis, is actually a key part of the healing program. Apoptotic neutrophils are cleared by macrophages, and that clearance event, called efferocytosis, is one of the signals that triggers the macrophage's transition from inflammatory mode to repair mode. Macrophages are the coordinators of tissue repair. But they are not static entities with a single fixed function. They are extraordinarily plastic, capable of adopting radically different behavioral profiles, depending on the signals they receive from the wound environment. This is called macrophage polarization, and it is one of the most important concepts in modern wound healing research. In the early inflammatory phase, macrophages adopt what researchers call an M1 phenotype. Driven by signals including interferon gamma, TNF-alpha, and bacterial products, M1 macrophages are inflammatory powerhouses. They produce large quantities of pro-inflammatory cytokines, IL-1 beta, IL-6, TNF-alpha, and reactive oxygen species that kill pathogens and amplify the clearance response. M1 macrophages are exactly what you need in the first hours to days after injury. They ensure the wound environment is cleared, sterilized, and primed for repair. Then the transition begins. As debris is cleared, as apoptotic neutrophils are consumed through efferocytosis, as interleukin-4 and interleukin-13 accumulate from mast cells and other early responders, the macrophage receives signals to shift its behavior. It transitions toward the M2 phenotype, anti-inflammatory, pro-repair, pro-resolution. M2 macrophages produce fundamentally different outputs from M1 macrophages. Instead of inflammatory cytokines, they release transforming growth factor beta, IL-10, and VEGF. Instead of reactive oxygen species, they promote collagen deposition and angiogenesis. Instead of amplifying the alarm, they begin suppressing it. The M1 to M2 transition is not a binary switch. It is a continuous spectrum, and the timing and completeness of this transition is one of the primary determinants of whether a wound heals cleanly or becomes chronic. When macrophages transition fully and on time, the repair phase begins efficiently. When the transition is impaired, when M1 macrophages remain dominant beyond their useful window, the wound environment stays inflammatory, fibroblasts cannot function properly, and the result is impaired healing or fibrosis. This macrophage polarization dynamic is an active target of therapeutic intervention. Several emerging treatments, including certain peptide approaches, are being studied specifically for their ability to influence the M1 to M2 transition timing. Cells cannot repair tissue alone. They need something to work on, a physical scaffold that gives them spatial organization, mechanical support,
and the biochemical cues they need to know where to go and what to build. That scaffold is the extracellular matrix. The ECM is not just a passive structural material. It is an active information system, storing growth factors, presenting signals to cells through surface receptors, regulating cell behavior through mechanical stiffness, and providing the three-dimensional architecture that cells use to orient their repair activity. In a healthy, uninjured tissue, the ECM is precisely organized. Collagen fibers aligned with the mechanical forces the tissue regularly experiences, growth factors sequestered within the matrix in a reservoir available for rapid release, and the whole structure maintained by a careful balance of synthesis and degradation. When injury disrupts this architecture, the ECM sends its own alarm. The fragments of collagen, hyaluronin, and fibronectin that result from matrix degradation, as we discussed in Part 2, activate PRRs on immune cells. But the intact ECM around the wound does something else. It releases the growth factors it had been storing. Within hours of injury, fibrin from the clotting cascade and fibronectin from disrupted matrix combine to form a provisional matrix in the wound space. This provisional matrix is not the final structure. It is a temporary scaffold, rough, disorganized, mechanically weak, but essential because it gives migrating cells something to grip and navigate through. Cells moving into the wound use the provisional matrix as their road surface. They extend cellular projections called lamellipodia that grip matrix proteins through integrin receptors, the same family of receptors that mediate immune cell adhesion to blood vessel walls. They pull themselves forward, remodel the matrix around them, and proceed toward the injury center. The mechanical stiffness of the matrix is itself an instructional signal. Softer matrices tend to direct stem cells toward neural or adipose lineages. Stiffer matrices direct them toward muscle or bone lineages. The ECM does not just support cells, it tells them what to become based on the physical properties of the environment they encounter. This is called mechanotransduction, the conversion of physical forces into biochemical signals. And it means that the biomechanical environment of a wound, how stiff the tissue is, how the matrix is organized, what forces are acting on it, is as important to healing as any chemical signal. With the scaffold in place, the alarm system coordinating immune response, and macrophages beginning their transition to repair mode, the body now needs to assemble the actual cellular construction crew. That assembly is coordinated by growth factors, and the sophistication of the growth factor network at a healing wound is genuinely extraordinary. Platelet-derived growth factor, PDGF, was among the first wound-healing growth factors identified. Platelets aggregate at the injury site within seconds of damage and release PDGF immediately from their granules. PDGF attracts fibroblasts and smooth muscle cells, stimulates their proliferation, and primes them to begin producing collagen. The timing is deliberate. PDGF arrives early, before the wound environment is ready for construction, to begin calling in the builders before they are immediately needed. By the time the inflammatory clearance phase winds down, a population of fibroblasts is already present at the wound periphery, ready to advance. Transforming growth factor beta, TGF-beta, is the most multifunctional molecule in repair biology. It stimulates fibroblasts to synthesize collagen and fibronectin. It promotes fibroblast differentiation into myofibroblasts, specialized cells that can contract the wound, pulling its edges together. It modulates immune cell behavior, and it regulates the production of matrix metalloproteinases, enzymes that remodel the ECM. TGF-beta is so central to the repair process that dysregulation of TGF-beta signaling in either direction, too much or too little, produces characteristic pathological outcomes. Excess TGF-beta activity is strongly associated with fibrosis, the excessive scar tissue formation that impairs function in conditions from liver cirrhosis to pulmonary fibrosis. Insufficient TGF-beta leads to impaired wound closure. Epidermal growth factor, EGF, and its receptor, EGFR,
drive the proliferation and migration of epithelial cells. When skin is wounded, it is EGF signaling that coordinates the migration of keratinocytes from the wound edges across the wound bed, the re-epithelialization process that closes the surface of a skin wound. Keratinocyte migration is not simply cells spreading randomly outward. It is a coordinated sheet movement. The cells at the wound edge migrate first, and the cells behind them follow, with the sheet maintaining its integrity through tight junctions between cells. The entire sheet moves together toward the wound center, guided by EGF and other growth factors. Fibroblast growth factor, FGF, promotes fibroblast proliferation and stimulates angiogenesis. Insulin-like growth factor, IGF-1, promotes cell survival and protein synthesis. Each growth factor contributes a specific piece of the construction program, and the interplay between them determines the architecture of the healed tissue. Every process we have described, immune cell function, fibroblast activity, collagen synthesis, stem cell differentiation, requires oxygen. And the injury that created the wound almost certainly disrupted the blood vessels that would have supplied it. Before construction can be completed, supply lines have to be built. Angiogenesis, the formation of new blood vessels from existing ones, is not just one component of tissue repair. It is the logistical prerequisite that makes all other repair possible beyond a certain tissue depth.
Vascular endothelial growth factor, VEGF, is the primary angiogenic signal. Released from hypoxic cells throughout the wound, it creates a concentration gradient that endothelial tip cells at the leading edge of vessel sprouts navigate toward. The tip cell extends filopodia, thin cellular projections that continuously sample VEGF concentration in the environment. The filopodium pointing in the direction of highest concentration grows longer. The cell body follows. The sprout elongates. Behind the tip cell, stalk cells form the wall of the new capillary. They proliferate, but do not explore. They fill in the growing tube while the tip cell navigates forward. When two sprouts from opposite sides of the wound approach each other and connect, a process called anastomosis, blood flow is established to the previously ischemic repair zone. Oxygen arrives, nutrient delivery resumes. Repair activity that had been limited by hypoxia can now proceed at full capacity. New vessel networks in healing tissue are initially disorganized, a chaotic tangle of capillaries that over time are remodeled and pruned into a functional vascular architecture matching the needs of the healed tissue. The connection between angiogenesis and the peptide biology discussed elsewhere in this series is direct. BPC-157's proposed mechanism centers on VEGFR2 signaling, the primary receptor pathway through which VEGF drives angiogenesis. If BPC-157 genuinely modulates VEGFR2 and eNOS activity in the ways the preclinical literature suggests, it is operating precisely at this step, the step where blood supply to the repair zone is being reestablished. The emergency response system we have described, DAMPs, PRRs, chemokines, neutrophils, macrophages, growth factors, angiogenesis, is extraordinarily powerful, which means it also needs an extraordinarily reliable off switch. Inflammation that does not resolve is not healing, it is damage. Chronic, unresolved inflammation drives some of the most prevalent diseases of modern life. Arthritis, cardiovascular disease, neurodegeneration, metabolic dysfunction, and accelerated biological aging. The off switch is not passive. It is not simply a matter of the inflammatory signals fading away. The resolution of inflammation is an active biological program with its own molecular mediators, its own signaling cascades, and its own precise timing. The specialized pro-resolving mediators, SPMs, are a class of lipid-derived signaling molecules first systematically characterized by researcher Charles Sirhan at Harvard. They include 4 families: resolvins, protectins, myrisins, and lipoxins. Each is derived from fatty acids, resolvins and protectins from EPA and DHA omega-3 fatty acids, myrisins from DHA, and lipoxins from arachidonic acid.
Unlike anti-inflammatory drugs, which suppress inflammatory signals, SPMs do something fundamentally different. They actively instruct the immune system to finish its work and stand down.
Resolvins tell neutrophils to stop migrating into the wound. They reduce neutrophil recruitment through chemokine receptors and promote neutrophil apoptosis, the programmed death that allows them to be cleared. Protectins protect neural tissue and promote the resolution of neuroinflammation, which is part of why omega-3 fatty acid research intersects so frequently with brain health. Murisins are produced by macrophages and are among the most potent stimulators of tissue regeneration signals identified in the SPM literature, promoting fibroblast proliferation and the regenerative capacity of the wound. The repair tissue itself provides feedback that drives resolution. As collagen scaffold forms, oxygen levels rise, reducing HIF-1A signaling and the VEGF production it drives. As debris is cleared, damp concentrations fall, reducing PRR activation. As the tissue architecture is restored, the mechanical distress signals disappear. Resolution is a closed loop. The damage launches the response. The response repairs the damage. The repaired damage removes the signal. The removed signal resolves the response. And in healthy tissue with functional SPM production, the whole system returns to the baseline surveillance state it was in before the injury occurred. Chronic inflammation is one of the most discussed concepts in modern health. It is implicated in heart disease, cancer, Alzheimer's disease, type 2 diabetes, autoimmune conditions, and the general process of biological aging. Understanding what it actually is at the molecular level requires going back to everything we have covered. Chronic inflammation is not simply a lot of inflammation. It is inflammation that has failed to resolve. In acute inflammation, the appropriate self-limiting response to injury, the entire sequence we have described runs forward, completes its job, and then resolves. The alarm turns off. The immune cells clear out or die. The tissue returns to homeostasis. In chronic inflammation, one or more nodes in the resolution cascade have failed. The most common failure points are: impaired SPM production is particularly significant. SPMs are synthesized from dietary omega-3 fatty acids, specifically EPA and DHA. Populations with low omega-3 intake have measurably impaired SPM production. Some researchers argue that the epidemic of chronic inflammatory disease in modern populations is partly a consequence of dietary omega-3 deficiency, specifically the inability to produce adequate specialized pro-resolving mediators to complete the resolution program. This is why dietary omega-3 supplementation shows consistent anti-inflammatory effects in human trials, not because fish oil suppresses inflammation, but because it provides the substrate for the active resolution program the body runs naturally. This distinction has direct implications for how chronic inflammatory conditions are best approached. Suppressing the inflammatory response with anti-inflammatory drugs, NSAIDs, corticosteroids, reduces symptoms by blocking the alarm signal, but it does not complete the resolution program. In some contexts, prolonged anti-inflammatory drug use can actually impair healing by blocking prostaglandins and other lipid mediators that are needed in the early phases of the repair response. The more complete therapeutic approach, and the direction much current research is moving, involves supporting the active resolution of inflammation, rather than simply suppressing it. Growth factor therapies, including bone morphogenetic proteins and platelet-rich plasma, operate at the growth factor broadcast node. They supplement or amplify the molecular signals that direct cellular construction activity. Their clinical evidence is strongest where the natural growth factor gradient is inadequate, impaired healing in elderly patients, avascular tissue, large tissue defects. Stem cell therapies operate at the recruitment node, providing additional repair cells to a wound environment that may not have sufficient endogenous stem cells to draw from. The SDF1/CXCR4 pathway we discussed is a primary target of interest for improving stem cell homing to injury sites. Antifibrotic therapies target the resolution node. specifically the TGF-beta signaling that, when chronically activated, drives excessive collagen deposition and impaired tissue architecture.
Peptide-based therapies, the subject of our other videos in this series, occupy multiple nodes simultaneously. BPC-157's proposed activity at VEGFR2 and ENOS addresses the angiogenesis node. Its proposed effects on fibroblast behavior address the ECM and growth factor nodes. TB500's actin sequestration mechanism addresses the cell migration node, the step where repair cells physically travel through the wound toward the damage. The biological logic of these compounds becomes significantly more comprehensible when you understand the system they are attempting to work with. Your body does not guess where the damage is. It does not wait for conscious direction. It does not require instruction. From the millisecond a cell is damaged, calcium waves propagate, damps escape, pattern recognition receptors engage, chemokine gradients form, immune cells navigate, macrophages coordinate, growth factors recruit, scaffolds assemble, blood vessels grow, and specialized mediators bring the entire response to an organized close. All of it, simultaneously, continuously, without you ever knowing it is happening. That system has been operating in every human being since before recorded history. It is one of the most sophisticated coordination systems in all of biology, and it is running inside you right now. Understanding it does not just make injury biology make sense. It gives you a lens for evaluating every therapy, every supplement, every recovery strategy, and every emerging treatment in regenerative medicine, because all of them are, at some level, speaking the language this system already knows. Subscribe to What the Health for the science that lets you evaluate these things properly. New video every week.